I think it's pretty solid advice.
Groups of cells in different parts of your brain, they're all connected.
The statistics just don't back it up. There's nothing here.
Any complex life you find has mitochondria.
We have discovered things from athletes juicing.
My name is Matt Kaberlein, and welcome to the OptiSpan podcast. Hey everyone, welcome to the OptiSpan podcast. On today's episode, I'm going to go over some questions that came out of a webinar that I participated in a little more than a week ago about the results of the Pearl trial. So this is a webinar that was put on by Ageless RX. It's the entire webinar is available on their YouTube channel. We'll put the link below. I'd encourage you to go check that out. And we had a question and answer period after the presentation of the results that went on for about half an hour, but there were quite a few questions that we didn't get to. And so I'm going to go through several of those. I think I have 17 of them here today. So these are questions that were submitted by participants in the trial. I wasn't actually able to get the questions from the webinar chat. So if any of you asked questions in that webinar or you've got questions after this that you would like to see answered, please put them in the comments section below. We'll do another AMA episode to address any questions that we haven't gotten to. Again, as always, this is just my interpretation. A couple of disclaimers. So the Pearl trial itself was sponsored by Ageless RX. Ageless RX is a for-profit company in the telehealth longevity space. I have no formal relationship with Ageless RX, nor do I have any financial interest in Ageless RX. I also, unfortunately, have no financial interest in Rapamycin. If there were an index fund or something like that for Rapamycin, I'd probably be an investor. But I know there are people out there who seem to think that I've got, somehow I'm going to make money off of Rapamycin. And at least at this point, unfortunately, that does not seem to be the case. So I have no financial conflicts of interest for the topic that we're going to talk about today. Okay, so the Pearl trial stands for Participatory Evaluation of Aging with Rapamycin for Longevity. It was a 48-week double-blinded, randomized, placebo-controlled, what's called a decentralized study, meaning this was done, at multiple locations. Really, these were people participating in the trial at their home and getting blood draws periodically. So it's decentralized, meaning there aren't a small number of study sites. And the goal here, as stated by the sponsors, was to evaluate the safety and efficacy of rapamycin in mitigating clinical signs of aging in a normative aging cohort. So the initial results of the study have been posted in a preprint, not yet peer-reviewed, but you can take a look at the preprint that is available on medRxiv. Again, we'll put a link below to the preprint for those of you who are interested. And then, as I mentioned, the webinar, Dr. Zazala went through the data from the preprint, and then several of us participated in a discussion afterwards. And that video, as I mentioned, is available. You should check it out. So I want to make a few important points before I get into some of the questions that were left from the webinar. First, in my opinion, this trial is not statistically powered for efficacy, meaning that it was not designed to be an efficacy trial. So despite the sponsors saying that one of the goals was to evaluate efficacy, this trial had no statistical likelihood of being able to demonstrate efficacy. That's important to understand. So there were only 115 participants in the trial overall. They were split up into three groups. So a 10 milligram group of rapamycin, a 5 milligram group, that's once weekly, of rapamycin, and then a placebo group, 40 in the 10 mg, 36 in the 5 mg, 39 in the placebo. So very small cohort sizes in order to detect efficacy. And as far as I know, no formal power calculation really was done to establish statistical power. So this is really a safety trial with the hope going in that maybe we will see some hints of efficacy. But that was never like a likelihood that they were going to be sufficiently powered to actually accomplish that. So another thing that's important to appreciate, and we'll talk about this a little bit more, is that the study design used compounded rapamycin. So that means that the drug was custom formulated at a dedicated compounding pharmacy from raw ingredients. as opposed to being produced at a pharmaceutical manufacturer. So compounding is a more complicated topic, probably worth a dedicated episode, but as we'll see, one of the consequences in this case of using the compounded pharmacy was that the formulation used in Pearl only had about 29% of the bioavailability of the generic sirolimus tablets. So sirolimus and rapamycin are the same molecule, or the Rapamycin tablets. So again, just important to appreciate that's, a little bit less than 1/3 of the bioavailability that you would normally get or that they anticipated when they set the dosing for the trial. So the target dose was 5 milligrams in one group per week, 10 milligrams in the other group per week. The actual effective dose was more like 1.5 to 3 milligrams if they had been using the tablets as opposed to compounded rapamycin. So that's important to appreciate as we start to think about what does this mean in terms of dosing and pharmacokinetics and things like that. And the Ageless RX group put out a preprint on this bioavailability of the compounded rapamycin, also on medRxiv. We will also put a link to that in the show notes. So what did we learn from the Pearl trial, sort of overall big picture? I already mentioned the compounded rapamycin complications. So at this dose, which again was about 1/3 the dose they anticipated, there were no side effects over the time frame of the study. That's good. And there was at least some decent evidence, in my view, for improvements in body composition, particularly preservation of lean mass in women taking rapamycin. There were some more speculative potential benefits in terms of perceived quality of life. And I think other than that, there wasn't a lot that we can hang our hats on from this study. So still think it's an important study. I think it did provide further evidence for potential benefits. And again, additional, I think, confidence around the lack of side effects, at least with once weekly dosing, again, at the doses that were used in this study. So, like I said, encourage you to check out the full video of the webinar where Dr. Z and members of the panel, including myself, go into more details. Okay, so let's dive into some of your questions. So question number 1, can you talk more about the compounded versus generic/commercial rapamycin? So I already talked about the relative bioavailability of this formulation of compounded rapamycin. I think the only other thing that I would say here is if you are getting and or using compounded rapamycin, and this is probably true for many other things that can be compounded, pharmaceuticals or other components that you can get compounded, you just want to be cautious about the equivalency of the bioavailability or dosing. And, some compounding pharmacies also have gotten into trouble because they don't always put what you think is going to be in the medication in there at the amount that's supposed to be in there. So just need to be a little bit careful with compounding. Much cheaper in most cases. So there's also a lot of value to compounding pharmacies. In specifically the case of rapamycin, though, I think it's important to understand that other compounded formulations may have higher or lower bioavailability than the compounded formulation used in Pearl. That's going to depend on, at least in part, the encapsulation method and product used. So at least one of the major reasons why compounded rapamycin has lower bioavailability than the tablets is because rapamycin is unstable at stomach pH. And so you can do a variety of things to get the rapamycin past the stomach into the small intestine where it can be released and taken up with better bioavailability, including using enteric capsulation formulations, which will have different efficacy in terms of getting the rapamycin through the stomach acid environment. So just appreciate other compounded formulations may have better bioavailability. Some may have lower bioavailability. Really, the only way to know is to measure it in blood at specific time points after dosing. Okay. Which brings me to question number 2. What is the most effective way to check rapamycin blood level? So I don't know what the most effective way is. I am familiar with the LabCorp Sirolimus blood test. It is quantitative, I think, down to about 1 nanogram per mil. So that's the sensitivity of the assay. So once you get below that, they won't return a value. But I think it's good enough for the uses that most people who are taking rapamycin off-label or most clinicians would want to use it for. So that blood test is available. It's not super expensive. We're actually working on ways to make some of these blood tests that we think are important more easily available to people. So stay tuned for that. But for now, you can get your doc to give you a requisition for the LabCorp blood test, and that should be one good way to measure rapamycin blood levels. Okay, question number 3. Given the less significant results for males, can you speculate on what dosage would be more appropriate for men, e.g. 10 milligrams, 15 milligrams, etc., or change dosing frequency, e.g. once every 2 weeks, assuming a larger dose? Okay, so this is referring to something that came out in the preprint and in the webinar that the hints of efficacy, the best evidence for efficacy for body composition were seen in women, not in men. This led to some speculation that women are going to be more sensitive at a given dose of rapamycin on average. Of course, there's huge inter-individual variability. So again, I'll come back to this probably, but I want to be cautious not to overinterpret the speculation about women being more sensitive to men. I think this is an arrow pointing in that direction, but hardly definitive in my mind based on the results of this one study. So the other thing I will say, again, is keep in mind that the formulation here was compounded. So the actual effective dose was far lower than the dose they were going for, the 5 and 10 milligrams if you were taking generic Everolimus tablets or Rapamycin. So given that, at this point, I don't see a lot of rationale for deviating too far from what is more or less the standard or the most common doses being used off-label, which seem to fall in the 6 milligrams once per week, plus or minus 4 milligrams. Again, this is speculation in the absence of data. We really don't know where the optimum is. And this question sort of alluded to maybe dosing once every 2 weeks as opposed to once every week. Then you can get a higher acute dose and longer time for trough levels to come down to 0. I'll come back to that concept in a minute. Again, we just don't know. So that might be a good strategy. It might not. I wish I could do better than that. At this point, I don't see any evidence to make me believe, though, that the 6 plus or minus 4 is too far off from where it makes the most sense to land. And I would suggest that actually measuring rapamycin blood levels, because of the individual variability in bioavailability and pharmacokinetics, is probably as important, if not more important, than the absolute dose that you are taking when you're trying rapamycin off-label. So question number 4, is there any suggestion of sexual dimorphism in the context of longevity in humans? Okay, so a couple things to say here. I'm not sure if this is the question being asked, or at least the intent of the question. As phrased, the answer is yes. There is clearly sexual dimorphism in terms of longevity in humans. Women live longer than men. That much is clear on average. Whether or not health span differs substantially between the sexes, I think is still a little bit more of an open question. Is it the case that health span is longer in women, shorter in women, the same in women and men? It kind of depends on what trait you're looking at. So I'm not going to wade into that here. That's a longer conversation. But certainly for longevity, there is sexual dimorphism. Now, for rapamycin in particular, as I already alluded to, this study, the Pearl study, provides, I think, some initial indications that there may be a difference in the sense that females, on average, again, may be more responsive to rapamycin at lower doses than males. Again, this is just the first indication. It is far from definitive. I want to caution people not to overinterpret this result to assume that rapamycin is more effective in women than in men. This is just a first hint that maybe that's the case. Certainly in mice, at a given dose, female mice, if they're C57 Black 6 or UM Het3 mice, seem to respond to rapamycin better than males do at a given dose. But if you push the dose far enough, the males will catch up, and you can potentially even achieve a dose where the males are optimized for lifespan, and that's too far for females. So it's not, as far as we can tell, the case, even in mice, that rapamycin works better for females than in males. It's just that the dose response for optimal dosing is maybe shifted a little bit in females versus males. And that's not even clear that that's the case across, a bunch of different genetic backgrounds. There's probably going to be a genetic component to this as well. So as with all things biology, it's complicated. And I suspect, again, that the individual variation is going to be more important than the sex component here for identifying optimal doses for rapamycin in terms of potential effects for health span metrics and longevity. Okay. So the next question is a long one, a question that is controversial and perhaps a bit off topic, but on everyone's mind. Is RapaAUC, that's area under the curve, or peak? So we're talking about now the pharmacokinetics of rapamycin, the peak being the highest value achieved, the area under the curve being, if you think about rapamycin going up, hits the peak, and then it comes back down over a period of days, the total area under that rapamycin curve. So is the AUC or the peak more important for longevity and immune function impact of rapamycin? So should a person aim for a high peak, but a short residence time clears quickly to reduce side effects? And you would do that by taking a higher dose of rapamycin, potentially on an empty stomach, maybe. Or should a person aim for slow absorption and long residence time at the cost of lower peak RAPA in blood? So this is a great question. With the answer completely unclear at this point. Again, we're working in the absence of data to really give us a definitive feel for what the likely right answer is. So first of all, I'm not sure, given that, why this would be controversial at all, because there's no compelling evidence really one way or the other, at least in my view. So if people have strongly held opinions on this, in my view, they're just arguing from a position of ignorance because there really isn't any data to strongly support one side or the other. Having said that, my guess, again, in the absence of data, or at least a lot of data, is probably that peak level is more important than area under the curve. And one reason why I say that is that we know that tissue distributions for rapamycin are going to be different, meaning different tissues will have different access to the drug when you dose. And like, for example, the liver probably sees strong mTOR inhibition when you dose with rapamycin, whereas if you're taking a low dose, the brain might not see any mTOR inhibition. So different tissues are going to be differentially sensitive to a given dose of rapamycin. And so if we believe that the effects of rapamycin, and I do, are mediated by effects on more than one tissue, direct effects, then a higher acute dose would be more likely to see some level of mTOR inhibition, broadly speaking, across tissues. The alternative explanation could be that all of the effects of rapamycin are immune-mediated, and we really don't need high doses. So again, speculation in the absence of data, but I think that's a reasonable speculation. So that's one reason why I would guess that acute dose is more important than AUC or peak dose. And then the other sort of piece of information here that I think is relevant is, at least so far, we don't really see much evidence for bad effects associated with high acute exposure to rapamycin. So for example, there are anecdotal reports of people taking dozens of milligrams of rapamycin. There are even a couple of case reports of people taking more than 100 milligrams of rapamycin once acutely without any real negative effects. I think one person got high triglycerides for a little while after taking more than 100 milligrams of rapamycin. So there doesn't seem to be much in the way of real risk. And again, I'm not suggesting anybody go out and take 100 milligrams of rapamycin. Okay, don't do it. But there's not much reason to be super worried about really dangerous side effects from a one-time high-dose rapamycin or even repeated acute doses of rapamycin, as long as you allow the rapamycin levels to clear out and the mTOR activity to return to normal in between dosing. So that's sort of my intuition, but again, very, very speculative. The flip side of that, of course, is that we do know that chronic low-dose rapamycin, just a few mgs every day, does lead to some significant side effects, or at least a higher side effect profile than we see from once weekly dosing of rapamycin. So, all very speculative, but that's my guess for now, is that the peak is probably more important than the AUC for efficacy. Who knows? Okay, next question. Perhaps mRNA delivery could be used in the future to inhibit the correct part of the mTOR pathway. I know some folks are doing that for senolytics. So absolutely, that's absolutely true. I think there are a variety of different ways that we could think about targeting the mTOR pathway, or really probably more correctly, the network that mTOR acts within. I think the challenge here is we don't know what the right target is. So even if we had tools to very specifically target a single component of the network or a couple of components of the network, we don't really know where to target to give us better efficacy than rapamycin and or fewer side effects. So in the absence of that information, yeah, it would be it's a great opportunity, but I think we really need to know what the right things to target are before we could really use things like mRNA approaches or other approaches to specifically knock down components of the pathway or the network. Question number 7. What do you believe as to the reasonableness of a dosing schedule of 8 weeks on and 4 weeks off Rapa? Current advocates include Peter Attia. Would this negate the positive benefits along with recalibration of, e.g., insulin for overactivation of mTORC2? Okay, so this person is specifically asking about a 8-week on rapamycin, 4 weeks off break, 8 weeks on, 4 weeks off. I'm guessing that's what it sounds like. A few things to say, and honestly, I don't know if Peter has advocated this or not, so maybe. But I think in general, a couple things I would say. One is the cycling of rapamycin, which I've talked about. That's what I do. I don't do this particular cycle, 8 on, 4 off, but I do cycle rapamycin. I think there's reasons to view that approach as potentially a good alternative to continuous dosing. So we know from studies in mice that somewhere between 4 and 12 weeks of treatment with rapamycin is long enough to see decreases in inflammation across many, many tissues and improvements in function across at least a subset of tissues and lifespan extension. So a single 12-week treatment with rapamycin increases lifespan, and then others, including, I think, Dudley Lamming's lab, have shown that different types of intermittent rapamycin dosing can also increase lifespan in mice. So in mice, cycling works, and that time frame of 4 to 12 weeks on is enough to give benefits at the functional and molecular level across different tissues and organs. Some evidence in dogs for improvements in heart function in that time frame, and some evidence in people for improvements in immune function in a 6-week time frame. So it seems likely that you can get at least the anti-inflammatory benefits of rapamycin in a 4 to 12 week period, depending on dose and obviously probably depending on individual parameters. Is it as good as continuous in terms of efficacy? Who knows? In mice, my intuition is it's probably not quite as good as continuous dosing. If we were to optimize both, my guess is you could get a little bit better effect from continuous dosing for longevity than you could from a single transient dose or intermittent dosing. But again, that's speculation. In people where we're probably more concerned about potential side effects, particularly risk of things like bacterial infection because of the complex environment that we live in, there may be some benefits to this sort of cycling on and off. So again, my personal view right now is that cycling probably makes sense. Again, I very much believe that at least many of the anti-inflammatory effects of rapamycin set in within a couple of weeks. And, again, depending on the dose, by 6 to 12 weeks, you've probably sort of optimized the anti-inflammatory part. And it's going to take a little while if you stop taking rapamycin for that inflammatory signaling to come back up. And there may be some benefits to stopping rapamycin for a while and allowing mTOR to sort of re-establish homeostasis. But we're working in the absence of data at this point. So that's kind of my best guess. Okay. Question number 8. Considering the variable pharmacokinetics of rapamycin metabolism among individuals, what do you believe should be the target blood levels of rapamycin for those taking it weekly with the goal of optimizing longevity benefits while minimizing potential risk? Okay, so again, we really need the controlled studies that are powered for efficacy to answer this. So this is where something like the Pearl trial, but larger and well-powered for a specific efficacy endpoint, would be really, really useful. We don't have that, obviously. So again, my current speculation, given the limited data available, is that we probably want to try to achieve the highest dose that allows for at least a couple of days where the trough level of rapamycin is below the detection limit of the assay. And so what I mean by that is if the detection limit of the assay is, say, 1 nanogram per milliliter in blood, you take the dose of rapamycin on day 0, right? You're going to get a spike after a few hours, and then it's going to come down. And again, I went through the kinetics in a fair amount of detail a couple of times on the podcast, so we'll link to those and you can check those out. But depending on the dose you take and your personal rate of clearance of rapamycin and probably other things as well, you may come down and be below the detection limit after 24 hours. You may not get below the detection limit by the end of the week before you dose again. So this is total speculation, but my total speculation right now is I would shoot for being below the detection limit of the assay for at least a couple of days before that next dose. Now, if you're cycling and doing, 8 weeks on, 4 weeks off, maybe you don't need to get below the detection limit for that next dose because you're going to have a period without rapamycin for a while. But again, given what we know right now, that would be sort of what I would target personally. Again, not medical advice. This is just what I would do. My personal opinion is try to take a dose that will make sure you're below that detection limit for at least a few days before the next dose. Now, do you want to try to go as high as you possibly can to achieve that? I would say that depends on whether you experience side effects at that dose when you go as high as you possibly can to achieve that. If you're getting mouth sores and they're bothering you, maybe you want to back off. I personally have never had any issues with mouth sores, so that hasn't been something that I've been concerned about. Okay, so that was number 8. Let's go for number 9. I've read that Everolimus is less specific to mTOR2, so mTOR complex 2, and has more effect on mTOR complex 1 than generic rapamycin. Is this true? And if so, does it make sense to prefer Everolimus if excluding financial reasons? Okay, so again, just for those of you who maybe aren't rapalog aficionados, Everolimus is a derivative of rapamycin. It's called a rapalog. Biochemically, Everolimus works just like rapamycin. And so what this question is asking, though, is it a more specific inhibitor of mTOR complex 1 than rapamycin? And again, most of you probably are familiar with this idea, but there is a model that's been put out there that chronic treatment with rapamycin inhibits mTOR complex 2 indirectly, and that some of the side effects of rapamycin at least are due to inhibition of mTOR complex 2, whereas the benefits of rapamycin, at least for longevity and health span, are due to inhibition of mTOR complex 1. Okay, let me clarify that statement. It is the case this has been seen in many, many studies. It is the case that chronic treatment of high-dose rapamycin, both in cell culture and in vivo, meaning in tissues of animals, will lead to inhibition of mTOR complex 2 through indirect mechanisms. That does happen. The model is that it's that inhibition of mTOR complex 2 that's really driving most of the side effects of rapamycin treatment, and or that all of the benefits are due to inhibition of mTOR complex 1. Data to support that, but it is, I would say, an incomplete model. And I don't think it's actually completely correct. I think it's more complicated than that, my personal view. So here the question is, if we had a molecule, and maybe it's Everolimus, that did the good stuff that rapamycin does for mTOR complex 1 without doing the bad stuff that rapamycin does for mTOR complex 2, assuming that it really is bad stuff, should we take it? And is there any evidence to believe that that's the case for Everolimus? There's a little bit of evidence to believe that's the case for Everolimus. A couple of papers have been published suggesting that treatment with Everolimus is has less of an effect on mTOR complex 2. So there was a study published in Blood, I think in 2015, and some work from the Lamming lab where they looked at both Everolimus and another rapalog called temsirolimus. Both of those studies provided some suggestion that the other rapalogs were less effective at inhibiting mTOR complex 2 with chronic treatment. They all looked at a limited number of biomarkers of mTOR complex 2. It was not an apples to apples comparison in terms of, at least it wasn't clearly shown to be an apples to apples comparison in terms of the amount of drug given and the total inhibition of mTOR complex 1. It's not really a criticism of those studies because that's hard to do, but it's just worth appreciating that there are reasons to think that it's not as clear as it was claimed to be in those studies that the other rapalogs are, in fact, less mTOR2 inhibitory. And probably most importantly, very, very limited evidence yet that Everolimus, temsirolimus, or other rapalogs can, in fact, have benefits for longevity or health span in animals comparable to or better than rapamycin. And to me, that's really the crux of the situation. In the absence of that evidence, it's really hard to make a recommendation that Everolimus is a better choice than rapamycin. Having said that, in people where we really have a lack of data, you could make the case that the best evidence for benefits in terms of at least rigor of the clinical trial comes from a study of Everolimus that Joan Manick did, where they showed that 6 weeks of treatment with Everolimus was sufficient to improve flu vaccine response in healthy elderly people. So there is some evidence in humans that Everolimus can at least have a benefit in terms of that one age-related functional decline. So again, my sort of take home is, if you want to take Everolimus, take Everolimus. What's the right dose there? I think you have to be guided by the Manick study. That those studies suggested that 5 milligrams once a week was sufficient to boost flu vaccine response with side effects that weren't really much different than the placebo group. So if you want to take Everolimus, take Everolimus. Again, biochemically, pretty identical mechanisms of action. I don't see a lot of rationale for believing that Everolimus is going to be superior to rapamycin, or that the side effect profile is going to be fundamentally different at this point, especially with the once weekly dosing. Okay. Question number 10 is another rapalog question related to the Everolimus question. Are any rapalogs looking more promising than others, or is it too soon? So again, there was a lot of excitement around the idea of identifying mTORC1 specific rapalogs a few years ago. I haven't really followed this in a lot of detail, so it's possible I've missed something new here, but I haven't seen anything recently that really looks compelling to me in terms of the idea that we've got a better rapalog or better rapalogs out there. There was a paper from Brian Kennedy and Dudley Lamming. I want to say it was like 2019 maybe, so it's been several years ago, that talked about an mTOR complex 1 specific inhibitor and had some evidence for mTORC1 specificity, but really nothing in terms of longevity or health span benefits. And I don't know why it's taking so long to move this stuff forward, but I really haven't seen anything in this space to make me think that there's any any really novel and better rapalog in the pipeline, but I know there are people working on it, and maybe there's data out there that I'm just not aware of. Also, probably worth mentioning, there are other ways to inhibit mTOR that are different from rapalogs. So I've talked about this before on the podcast, but rapalogs have a very specific biochemical mechanism of action. They are what are called allosteric inhibitors of mTOR. They bind another protein, FKBP12, and it's that complex that inhibits mTOR. There are also direct catalytic or ATP competitive inhibitors of mTOR, and Aura Biomedical, which is a company that we spun out of my lab at the University of Washington. So I do have a financial conflict of interest with Aura Biomedical. Aura Biomedical has identified a different type of mTOR inhibitor that, at least in C. elegans, has about twice the effect as rapamycin on lifespan. So I have no doubt that there are better mTOR inhibitors out there in terms of their ability to affect longevity or health span. I just haven't seen any data yet in mammals that is compelling to demonstrate that. Okay. So next question, have you considered an injectable form of rapamycin as opposed to the ingestible form, the oral form? So first of all, there are both tablets and liquid oral forms of rapamycin. There is also a topical form of rapamycin. To the best of my knowledge, there is no clinically used injectable form of rapamycin, although some of the mouse studies that have been published, including work from my lab, have used IP injections of rapamycin daily or every other day. So certainly that is a reasonable method of delivering the drug, and plenty of evidence for health span and lifespan benefits in laboratory mice from injection of rapamycin. It's a good question. I believe that if you wanted to develop an injectable form of rapamycin for human use, that would require a new FDA approval before it could go to market. I might be wrong about that, but I think it probably would. And so that's a pretty high bar for a drug that's off patent. As far as I know, nobody is developing an injectable form of rapamycin for any indication. Okay, question 12. I'm 48 years old and take Everolimus as a pulsed dose of 8 mg per week. The everyday aches and pains of getting out of bed, in and out of the truck, etc., completely vanished within 6 weeks of starting. I've heard that before. Has been life-changing. That's great. This seems to be a low-hanging fruit as a result for a trial that positively impacts quality of life issues for many people. Why don't more clinicians pay attention to this? Yeah, I agree. So it's frustrating. Yeah, there's a whole bunch of reasons why, not just clinicians, but basic researchers, clinical researchers, funders really ought to be paying more attention to this. So I think the answer ultimately comes down to somebody's got to pay for it, right? To do the trials, somebody's got to pay for it. And unfortunately, in our current system, there really is no incentive for a for-profit company to develop rapamycin for this kind of use, because the drug is off patent and just because of the way that drugs are regulated and the cost of doing FDA trials for a new indication are done. It just is not a reasonable thing for a for-profit company whose incentive is to make money to do that. And I mean, the fact is that while some individuals in these big pharma companies absolutely care, the companies are amoral, and it's not about helping people, it's about making money. So I don't know what to tell you other than somebody's got to step up and pay for the trials. And it's not only about clinicians paying attention, it's about actually having the resources to be able to do those clinical trials. So has been a constant source of frustration for me for more than 15 years. I don't know what to tell you, other than I wish it wasn't that way. Okay, next question. If we assume one of the reasons for minimal significant effects is low dosing, what are the techniques, strategies for dose finding? So this is referring, I think, to the Pearl trial in particular, that the reason why they didn't see efficacy is because of the low dose. I would say maybe, again, that trial wasn't powered for efficacy. anyways, do we just spend 50 years slowly increasing dosing in trial after trial until we start seeing negative effects / stop seeing positive effects? Or is there a better way to do dose finding in humans? So I think there's a couple of things to say here. Again, one of them is what I just said. Somebody's got to step up and put the money down. If someone put a few $100 million towards this problem, we could find out very quickly. So resources, again, are the rate limiting factor here, period. Having said that, I think the selection of endpoints plays a big role, and some understanding of the molecular mechanisms for how rapamycin might be impacting those endpoints would help with the dose finding, because if you had a hypothesis for how rapamycin was impacting, say, periodontal disease, which we do, you could then use molecular biomarkers along with non-invasive additional measures of efficacy to help guide that dose finding. So I think there are some strategies that can be used that can accelerate this. But again, it really comes down to somebody putting the money down to actually do the trials and accelerate the process. Otherwise, again, I hate to say it, I just don't see a path to where we're going to actually have definitive data anytime in the near future in the absence of those resources being allocated. Okay, we are on number 14. Is it a good idea to measure parameters like grip strength or lung capacity as additional indicators of improvement? Okay, so again, in the Pearl trial, the primary data that was reported on, at least so far, was body composition based on DEXA. And so the question is, would it make sense to look at functional measures like grip strength or lung capacity? Absolutely. I mean, I think in general, measuring functional and molecular biomarkers in combination is a good idea. Personally, I don't know that I would put grip strength or lung capacity near the top of my list for rapamycin, but for a trial like Pearl, that may make sense because they're easy for people to do at home. I personally put a bit more faith in comprehensive blood work. Like I said, body composition by DEXA, I think, is really good, and more sophisticated metrics of strength and muscle function that get beyond, grip strength and lung capacity. But having said that, yes, the answer is yes. I think we want a combination of functional metrics and molecular measures to really have more confidence about whether the intervention is achieving the efficacy that we're looking for. So there's a good question. Okay. Next question. Do you think that fasting for a day around taking rapamycin can make a positive difference, provided it may have an additive effect to mTOR and AMP kinase? So again, this makes perfect sense. Yes, fasting, certainly 24-hour fast will have an effect on mTOR. It will reduce mTOR signaling in general. So same direction as rapamycin. It will boost AMP kinase in general. So the question is it a good idea to combine a dose of rapamycin with fasting? And again, I don't know of any evidence really one way or the other here to really even speculate. So maybe the additivity for inhibition is valid speculation, is probably going to be true. That's going to depend to some extent on the dose of rapamycin you take and the degree of fasting, in the sense that, at some point you're going to floor mTOR, at least in key tissues like liver. So you may not, in fact, get additivity from a combination dose. And I don't really know what the effect of fasting would be on the clearance of rapamycin. So, if you were to alter the activity of the cytochrome P450 enzymes, for example, fasting itself may actually change the pharmacokinetics of rapamycin. So I think in the absence of that data, it's kind of hard to predict. And given that we spend a lot of time thinking about what is the optimal dosing of rapamycin, it's quite possible that fasting plus, say, 6 milligrams of rapamycin would actually push you over into the too much range, at least in terms of mTOR. So unfortunately, we just don't know at this point. And I hesitate. I don't have a strong feeling, so I hesitate to really even speculate in the absence of having a strong idea as to what the likely outcome would be. Okay. Question number 16. We're almost there, guys. What would the cost be to run a powerful long-term rapamycin trial, say 5 years, strong enough to power to be confident in results, including null results? Great question. And this, I think is really what it comes down to. This is what we would all really like to see. I think there are a few things to consider before you can even do a back of the envelope guess, which is what I will do, which it'll be totally back of the envelope. So don't hold me to these numbers. But I think before you even do that, you've really got to consider a few things. I think one thing to consider is, are you going to do this trial in humans or in companion animals? So pet dogs, for example. So that fundamentally changes what you can measure and the cost to measure it. Another question is, are you going for FDA approval or not? Because the FDA approval process is also going to increase the amount that it costs to do the clinical trial. So one goal could be just to determine efficacy, and you don't really care whether you're going to get a new label indication for rapamycin because doctors can already prescribe it, for example. And then I think the third thing to really consider is endpoint, because that's also going to impact the total cost of the trial. So let's just start with dogs, because I think that's relatively easy and I've got a pretty good feel for what it would take. So if you want to look at lifespan and health span, so something similar to what we're measuring in Triad, knowing what we know now, so you go back and not make some of the same mistakes, definitely you could do the definitive study, not just rapamycin, but pick your favorite indication, definitely for less than 10 million, probably less than 5 million, maybe even substantially less than that. So that's ballpark. Pick your favorite intervention, assuming the drug itself or whatever the intervention is doesn't cost millions of dollars. That's kind of what you're looking at. If you really want to do it right, make sure that you're going to get the answer at the end of the day. I think you can do it for 5 to 10 million for sure in companion animals. And that's looking at lifespan and health span metrics, multiple health span metrics as the endpoint. Super low-hanging fruit. Some people are moving this direction. It's a little surprising to me that more resources have not gone in this direction, given the potential impact and the market size for companion animals, if you can prove that you can slow biological aging in people's pets. But that's kind of what it would take. Okay. Humans, going to be more expensive. Just accept that off the bat. So if you start with a really inexpensive endpoint like periodontal disease, and you're not going for FDA approval, I'm guessing it wouldn't be a lot more expensive than the study I described in dogs. Again, 5 to 10 million, back of the envelope. I'm guessing you could do that definitive study for periodontal disease in that ballpark. That's assuming you're not trying to get FDA approval. If you want to get FDA approval, that adds a whole bunch of other bureaucratic stuff into the mix, and I'm guessing increases the cost 10x, right? By a factor of 10. But if you're not really interested in FDA approval, yeah, probably 5 to 10 million. If you're going to look at an expensive endpoint, let's say heart disease, I'm guessing it's 10 to 50x, something like periodontal disease. Total guess, back of the envelope calculation. And again, you can put that 10x multiplier on there if you're going after FDA approval. Now you start to see why it's really only the big pharma companies that are able to take drugs all the way through this full pipeline. And, if it's done at an academic institution, you can add another 50 to 100% in overhead costs to the budget of the project, because they're all going to ask for indirects. Okay, so I think this again illustrates the fundamental problem here. The entities who can afford to do the FDA level trials won't do them for rapamycin because it's off patent. NIH, at least so far, hasn't been willing to fund the well-powered trials. And what they have been willing to fund are what I would consider these unsatisfying phase 1-2 safety trials that are underpowered, but that let people publish a paper and get another grant and do another underpowered clinical trial. So it's super inefficient, but that's the model that, at least so far, for things like senolytics or metformin, that's what's happening. There are a bunch of these underpowered small clinical trials. And again, at least to date, groups like Evolution, which I'd say falls somewhere in the middle of the sort of, NIH academic and for-profit worlds, and at least in principle, have the resources to fund these studies that could actually move the needle, so far have been unable or unwilling to step up and really do anything significant in the space. And again, I'm talking about rapamycin, but I'm also talking more broadly about intervention testing in longevity. So there are opportunities here, guys. It would be nice to see some movement. Hopefully we'll get there. And again, I'll just point out, in my opinion, oral health in humans, longevity and health span in companion animals are really, really low-hanging fruit here. Hint, hint. Okay, enough said. Okay, last question. Given the resistance to reversing aging being an endpoint for trials, would restoration of function be a better option for these trials? Again, this is a good question. So the one thing I would say is I don't think that there's a resistance to reversing aging being an endpoint for the trials. It's that there is no quantitative way to measure reversal of aging. And if you want to, for an endpoint for a clinical trial, you have to have a quantitative endpoint that is going to be, if your goal is to get a drug approval or to convince the medical establishment or anybody really to adopt your intervention, it's got to not only be quantitative, but it's got to be believable. People have to believe that it's actually measuring something important. So where you want to go, and what FDA is certainly going to require, is a quantitative endpoint that is an accepted metric of either quality or quantity of life. So can you increase survival? That's unambiguous. Can you improve quality of life? There's some ambiguity there, but I think most of us have a pretty good feeling for what would make for an acceptable endpoint for an improvement in quality of life. And that's going to depend on the disease indication you're going after. If it's a disease, or in the context of aging, it's got to be something that clearly is beneficial for people's quality of life. So it's not that anybody's against reversing aging as the endpoint for a clinical trial. It's just we don't know how to measure it. There's no consensus for how to measure it. And so you can't really do a scientifically valid clinical trial for that endpoint. So the answer is yes. Functional measures are a better option for these trials, and particularly restoration of function in some cases can make sense. And again, I would suggest that really was the model that Joan Manick took when she was at Restore Bio, looking to see whether or not they could restore age-related declines in immune function. And they could. There was good evidence that they could. For a variety of reasons, they did not end up getting their drug through their pivotal phase 3 clinical trial. But I think that model of picking a smart endpoint where you can actually see an improvement in function, and you can convince the regulators that improvement of function is predictive for quality or quantity of life for at least a subset of people, is the right path for doing these sorts of geroscience clinical trials. And I think we'll see more clinical trials going that direction, in the next several years. And hopefully there can be some policy initiatives put in place to help accelerate those sorts of geroscience clinical trials at FDA. And that's something that I know people are working on, and I'm a huge supporter of, and I will keep trying to get out there and advocate for. That's it. Like I said, if we didn't hit your burning question, please put it in the comments section below, and we will definitely come back and do another episode on questions related to rapamycin. They can be directly from the Pearl trial or more general. So more to come on that. All right. Thanks everyone for watching this episode of the OptiSpan podcast. As always, please leave your questions and comments below. Please hit subscribe if you're not yet a subscriber, and I hope to see you next time on the podcast.